Welcome to our first semester cell biology course. Today, we are diving into one of the most fascinating and crucial processes in any living cell: nuclear membrane transport. Imagine the nucleus as a highly exclusive VIP club holding the cell's most precious information, the DNA. It cannot just let anyone walk in and out freely. We are going to explore exactly how the cell acts as a bouncer, checking passes and providing the energy needed to move vital molecules across this heavily guarded barrier. Let us get started. To understand how this transport works, it helps to imagine the nucleus as a highly exclusive VIP club located right in the middle of a bustling, crowded city. The city is the cytoplasm, filled with activity and floating molecules. Because the nucleus holds the DNA, it must be fiercely protected. It is surrounded by a thick, double-layered wall known as the nuclear envelope. If a molecule wants to enter or leave this VIP club, it cannot just sneak past the perimeter. It needs a specific, recognizable VIP pass. This strict regulation is what keeps the genetic code safe from chaos. Now that we have pictured our VIP club, let us look at the actual doors and the bouncers guarding them. The nuclear envelope is embedded with large, multi-protein channels called nuclear pore complexes. While tiny molecules and ions can slip through these pores passively, larger macromolecules like proteins and RNA cannot. They require active, facilitated transport. This is where soluble transport receptors, called karyopherins, come into play. Think of them as the VIP escorts. We have importins, which bind to cargo in the cytoplasm and bring it inside, and exportins, which carry cargo out of the nucleus. Without these escorts, large molecules are completely stuck. You might be wondering, why is this complex transport system so absolutely critical for a cell's survival? The answer lies in the central dogma of biology. DNA makes RNA, and RNA makes protein. When a gene is transcribed into messenger RNA inside the nucleus, that RNA is useless unless it is exported to the cytoplasm, where ribosomes are waiting to translate it. Conversely, all the machinery needed inside the nucleus, like DNA polymerases, are built in the cytoplasm and must be imported. If this bidirectional transport were to stop even for a moment, the cell could not express genes or divide, leading to immediate cellular death. Understanding the biological importance naturally leads us to ask how fast this process actually happens. Because nuclear transport relies on specific receptors binding to cargo molecules, the entire process behaves very much like an enzyme binding to a substrate. This means we can describe the rate at which cargo is transported using a mathematical model similar to Michaelis-Menten kinetics. By looking at the concentration of cargo in the cytoplasm, we can predict exactly how fast it gets moved into the nucleus. However, because there is a limited number of important receptors, the transport machinery can eventually become saturated if there is too much cargo. To put those kinetics into exact terms, let us look at the mathematical formula governing this movement. This equation comes directly from enzyme kinetics principles. Here, V represents the actual rate of nuclear transport, measured in molecules per second. Vmax stands for the maximum possible transport rate, which happens when every single important receptor is fully saturated with cargo. The bracketed C represents the concentration of the cargo molecule in the cytoplasm. Finally, Kt is the transport constant. It tells us the specific cargo concentration needed to reach exactly half of the maximum transport rate. If the cargo concentration increases, the transport rate V will also increase, but only up to a certain point before hitting that Vmax ceiling. Now that we have our formula, let us see this with a concrete example. Imagine a cell needs to import a specific transcription factor to respond to a stress signal. We are given a maximum transport rate of 120 molecules per second, a transport constant of 40 micromolar, and a current cargo concentration of 10 micromolar. We want to find the current rate of import. First, we substitute these known values into our transport equation. The numerator becomes 120 multiplied by 10, giving us 1,200. The denominator is 40 plus 10, which is 50. Dividing these gives us a final answer of 24 molecules per second. This means the cell is actively pumping 24 molecules every second, well below its maximum capacity. So, we have calculated how fast molecules move, but this brings us to an important question. What powers this movement? Moving molecules against their concentration gradient or organizing them so highly requires a massive amount of energy. Interestingly, in nuclear transport, this energy does not come directly from ATP. Instead, it comes from the hydrolysis of a similar molecule called GTP, which is mediated by a special protein called RAN. RAN exists in a high-energy state as RAN-GTP inside the nucleus and a lower energy state as RAN-GDP in the cytoplasm. The cell maintains a steep gradient of RAN-GTP across the nuclear membrane, and this gradient is exactly what dictates the irreversible direction of transport. To understand exactly how much power this RAN-GTP cycle provides, we turn to thermodynamics. This is the Gibbs free energy equation for the hydrolysis reaction. Delta G represents the actual change in free energy under cellular conditions, measured in kilojoules per mole. Delta G naught is the standard free energy change, a constant baseline value. R is the universal gas constant, and T is the absolute temperature in Kelvin. The fraction inside the natural logarithm represents the ratio of the cellular concentrations of the products over the reactants. When the concentration of GTP is very high relative to GDP, this logarithm becomes negative. This makes the overall delta G highly negative, meaning a massive amount of energy is released to forcefully drive transport. Let us apply this thermodynamic formula to a real human cell at normal body temperature. We're given a standard free energy change of negative 30.5 kilojoules per mole. The temperature is 310 Kelvin, and the gas constant is 0.00831. The cell maintains a product-to-reactant concentration ratio of 0.01. We plug all these values into our Gibbs equation. First, we multiply the gas constant by the temperature to get 2.576. The natural logarithm of 0.01 is negative 4.605. Multiplying those gives us negative 11.86. Adding this to our standard energy gives a final actual free energy change of negative 42.36 kilojoules per mole. This highly negative value confirms a massive energy release. Now that we have calculated the final energy, let us break down the mathematical reasoning to prove why this transport is strictly irreversible. We start by looking at the concentration ratio maintained by the cell. Because the cell keeps GTP levels much higher than GDP levels, this ratio is a very small fraction, specifically 0.01. When we take the natural logarithm of a number less than 1, the result is always negative. Here, it is negative 4.605. Multiplying this by the thermal energy factor yields a negative energy contribution of 11.86 kilojoules per mole. This step mathematically proves that the steep concentration gradient itself heavily favors the forward reaction. Continuing our proof, we now combine this gradient contribution with the baseline standard free energy. The standard free energy is already negative 30.5, indicating that GTP hydrolysis naturally releases energy. But when we add the negative 11.86 from the concentration gradient, the total delta G drops even further to negative 42.36. Because this final delta G is so far below zero, it proves that the reaction is highly exergonic and irreversible. This massive energy drop is what physically forces the important receptor to change its shape and release its cargo into the nucleus, ensuring that nuclear transport only flows in the correct, biologically necessary direction. Having proven the thermodynamics, let us visualize this entire process in action. On screen, you are looking at a detailed schematic of the nuclear membrane transport cycle. The double horizontal lines represent the nuclear envelope, separating the cytoplasm at the top from the nucleus at the bottom. Right in the center is the massive nuclear pore complex. Notice on the left how the important receptor binds to the cargo protein in the cytoplasm and escorts it through the pore. Once inside the nucleus, the high concentration of RAN-GTP binds to the important, forcing it to drop the cargo. The important is then carried back out to the cytoplasm, where GTP is hydrolyzed to GDP, releasing the important to begin the cycle all over again. We have covered a lot of ground today, so let us summarize the most important ideas. First, nuclear membrane transport is a highly secure, heavily guarded transit system, essential for the cell's survival. We learned that nuclear pore complexes act as the physical doors, while karyopherins, like importans, act as the VIP escorts. Second, we saw mathematically that the rate of this transport depends heavily on cargo concentration, much like enzyme kinetics. Finally, we proved that the steep gradient of the RAN-GTP cycle provides the massive, irreversible energy drop needed to drive this entire process. By carefully regulating what goes in and out, the cell keeps its DNA safe while allowing the vital flow of information required for life. Great job mastering this complex topic.